I'm going to be diving now into some pretty detailed analyses about using genetic approaches to monitor fish dispersal. So, a very different change in scope from the previous two presentations. We're talking about the worldview of the Nez Perce and the tribes in this area, and then a larger scale, comprehensive analysis of Columbia Basin tributary habitat. So, that's very humbling to then dive into these details. But one time at a festival, I had to give a stream ecology talk following a Cuban jazz band. So I'm going to be drawing on those skills to try to make this interesting. This was a very highly collaborative project. It was basically spearheaded by my master's student at Oregon State University, Kayla Kelly, but it involved a lot of different people that you see up there on the co-authorship list. But I want to call out specifically Joe Lemansky, who's in the room here, and Matt Kaler, who basically kind of pioneered this approach, and we'll be referring to that publication later. Um, but I want to start out by, let's see, how do I, down there? Okay. Yeah, I want to start out by just imagining, have you imagine a hypothetical watershed where you're doing a stream survey. So imagine you go out and you do a spawning survey. Okay, there you go. All right. You do a spawning survey, and those are locations of the adult reds that you find out on the river. And then you go back the next year, and the smaller icons are actually where you find the juveniles. But you don't have any information that connects where the juveniles came from in terms of their parentage. So you make some assumptions about what happened there. You see a few juveniles below those upper two adults that were found on a spawning ground, and then you see a bunch of juveniles below a red where there appears to be a smaller number of fish. So you might make some assumptions about that that include you may want to restore the spawning area, the spawning rearing habitat up in that area, or you might go to the next area and protect existing spawning habitat. Um, but then you say you do an analysis and you find out that you can identify the juveniles to those individual parents on the stream. So now the colors correspond with the familial relationship. So the yellow small dot is the juvenile from the adult in yellow that was found up there. And you would maybe make some different conservation decisions on the landscape. You might restore the spawning habitat quality in one area, or conserve and enhance the connectivity down in another area, or identify a location where fish are not traveling as much in between one area and they could have dispersed to. And so maybe you have a restoration project that would address connectivity. So, and then in a completely different scenario, I want to point out that all these locations haven't changed at all. We're still looking at all the same locations, but now the identity between the families has changed. So now you would look at doing something like restoring the spawning habitat quality in a lower area, but then talk about habitat complementation, or the overlap between spawning and rearing area in a lower area. And then you would conserve or enhance connectivity across the larger riverscape. And so these ideas point a lot to some of the literature and the ideas around habitat complementation, which is this idea that there are distinct and non-substitutable habitat used by different life stages. And Chinook are classic for looking into this because they have very complex life cycles and have different life history pathways. And this is really important for consideration of restoration and connectivity. So the restoration that a lot of us work on out here in the basin, and a lot of you actually implement, is meant to mitigate the habitat degradation, but it's very difficult to plan, implement, and to quantify, especially when it comes to asking the question, are the fish actually benefiting from the restoration? And so one of the questions that we ask are the target species dispersing to these restored habitats? And so the method that I'm going to talk about today really focuses on that dispersal question. As an aside, because we work out here in areas with hatchery supplementation, that has a lot of benefits, include aiding in recovering of the declining populations, but may have some drawbacks, including documented lower fitness in the hatchery origin fish. And then many of these populations exhibit intergression. So if you're interested in kind of either understanding the impacts of mixing hatchery stocks with natural origin stocks, or if you want to keep them separate, then these are important things to look at. So parentage-based tagging, this is where I do a lot of hand waving because I am not a geneticist, but I collaborate with geneticists. But we're basically genetically linking the offspring with the parents. And this is really important for this kind of a study because early life history studies are really difficult with fish that you can't tag at that very small stage. And so this approach is really ideal for looking at juvenile dispersal. So when you go out and you find juveniles, you take a fin clip and you're able to give them a parentage assignment. And three possible outcomes of that are you get a male assignment for the parent, you get a female assignment, or you get a double assignment. You can identify both of the male and the female parents. And if you just get the female assignment, you can identify the red location and the dispersal, so where that fish came from, assuming that the red was found at the carcass was found at the red. And I think in the Grand Ronde, that's a pretty safe assumption because river flows are so low during that time of year, and these fish are not moving around, and the carcasses are usually found right next to a visible red. If you if you get a match with a male parent, you don't get any dispersal data because those males are going all over the place in the basin. If you get a double assignment, you can look at the distance that the males move from the reds, but you can also identify the red location and the dispersal. And the kind of area of interest that I'm going to be talking about today is just those double assignments and the female assignments where we can address dispersal. But there's a lot of other really cool stuff that you can do with this data, including look at male movements, and talk to Joe Lemansky and others if you're interested in that subject. Um, so this paper that we published about a year ago, or maybe a little bit less, was spearheaded by Matt and Joe, and it was looking at watershed scale dispersal patterns over in the Middle Fork John Day. And so they kind of pioneered this specific approach. And but the distinct differences about the Middle Fork John Day is that's an all natural origin population, so you don't have that supplementation happening at the same time. And the kind of layout and the watershed context of that tributary is really different in terms of there's a lot of tributaries that fish can pop into and out of, and Catherine Creek is a really different kind of a stream without major tributaries. Um, so the objectives of Kayla's study were to determine where the fish disperse to through the riverscape, assess the difference in the dispersal patterns based on the spawner origin, so the hatchery versus natural origin, and to evaluate those patterns, including the dispersal distance, the direction, so up or downstream, and the use of restoration sites. Um, so the study area was Catherine Creek, which has been brought up a couple of times already today, and we've seen significant habitat restoration in that stream, although a lot of restoration as well. An important stream for Chinook spawning and rearing, and like I mentioned earlier, it has a supplemented population. So what I'm showing you now here are the locations in the first year of the study of the spawners. And basically all the spawning fish as they come up through Catherine Creek have to go through the weir, and which has a really high efficiency. So almost all the fish that come up through that weir get caught. And so you can get a genetic tag from those fish. And so in the summer of 2023, Kayla and her crews and a lot of other partners out there were doing the weir sampling, the spawning ground surveys that were kind of the traditional ODF and W spawning surveys, but then also went out there on a daily basis during the intense spawning surveys to get as many fish as possible. And what you're looking at are the blue dots are the natural origin spawners, and the brownish orangish dots are the hatchery. And so there was 143 carcasses for the genetic analysis. For the female origin identities, there were 77 hatchery and 13 natural. And then going out the next year and doing that summer par collection period in a standardized manner, and then also doing additional samples and sampling for abundance, and that was involving snorkel surveys and then extrapolating out to based on capture efficiency. So you basically have a picture of the where the adults are and then where the juveniles ended up, and then you get genetic samples from everybody, and then you use that information to find out where fish disperse from individual locations. Um, the restoration samples was a big part of this project because it was funded by the BOR, and they were really interested in looking at the efficacy of the restoration that's being done. And so there were 1,300 samples that were collected just in the restoration areas. The parentage analysis was spearheaded over in the Hagerman lab by the Critvic geneticist. And so Haley Nutzel was our primary collaborator on that. And so we use SNPs and retained individuals that had greater than 85% of the loci. And then a couple of different approaches were used for both the double assignment and the single assignment. So Snippet for the double and Sequoia for the single. And then the results of the parentage assignments were that most of the assignments, almost 60%, were both with the female and the male ID. About a third of them were a single female ID, and then a very small amount were a single male identification, and some of the fish that were caught were unassigned. So what you get and how you analyze that data once you have it is you can look at the dispersal distance of these different parentage groups. So we look at the median, the interquartile ranges, the confidence intervals, and then we look at the different parentage groups. So the table on the left is just looking at the female assignment, whether it's a natural origin or a hatchery origin. But on the right, we look at the double parent assignments, and that gives you a bunch of different groups according to whether the dam was a natural or a hatchery origin versus the sire. So you end up getting 6 different groups that you can analyze. And then the modeling approach, the data analysis there was a dispersal process model that was a multinomial regression that looked at the probability of an individual par that was dispersing to any given site based on whether it was in a restoration area, whether or how far or close it was to an individual red or other environmental factors. And then that was coupled with looking at the sampling process, which accounted for the probability of sampling a fish and getting a parentage-based tagging match, given it was present at a site. So all this was wrapped into the model. And then we used the Critvic model to account for unsampled fish that we didn't see. And so this was a Bayesian multinomial logistic regression, and some of the details of the model are there. I won't go into detail about that, but we basically had standardized sites with abundance, and we had 33 of those sites, but then we sampled outside of those standardized sites to get as many individual matches as we could, and you can see that we had almost 1,500 fish that had a genetic match. Okay, jumping into kind of the data summary here. So this is basically a generalized result when you look at the dispersal distribution by the female origin. So it's not related to where the fish spawned in the basin. It's just basically looking at how far did each individual fish distribute from the red. And you can see that most of the dispersal is downstream, so not a huge surprise there. So about 92% of the par went downstream, but some of them actually went upstream, of a much smaller percentage. And when you break that down by natural origin versus hatchery origin, you actually don't see that much of a difference in the results, but you do see a little bit more upstream dispersal from the natural origin fish. But keep in mind that we had way fewer sample sizes for the natural origin fish. So there's about 400 of those, and we had a lot more fish that dispersed downstream. Um, so now I'm going to show you a different set of graphs that kind of show the information in a little bit different way. So this is getting at, according to where in the basin fish actually spawned, where did fish disperse to? So now, rather than just an overall generalized approach, we're looking at the gray bar represents where the fish spawned. Right there, it's centered around river kilometer 35. And then the blue for natural origin and the brown for the hatchery origins shows the relative dispersal probability downstream. So you can see some of the fish ended up actually in the spawning area, and those were mostly the natural origin fish, but a lot of the fish ended up downstream of where they were. And this specific site was the Catherine Creek 44 restoration project. So I'm going to show you a bunch of plots there that show different parts of the basin. So from top to bottom, you're looking at the restoration to Badger Flats to the restoration site, the Catherine Creek State Park, Hall Ranch, which is a spawning area, and then upstream at the acclimation site to the forks, and then up into the South Fork tributary. What these graphs show are the relative dispersal patterns of the hatchery and the natural origin, but they're not showing the amplitude because they're all scaled down to relative density. So it's important to note that like there was a lot more fish that were spawning in the Hall Ranch than a lot of other areas in the basin. A couple of things to take away from a graph like this is the red circle is basically showing that a lot of the fish, both hatchery and natural origin, are migrating from the spawning locations to the CC 44 restoration area. So they end up traveling and traveling, and then when they hit that restoration area, they stay there into that summer par rearing period. Um, but then when you get far enough from that area, you get fish that are actually hanging out in or near the Catherine Creek State Park area or the Hall Ranch area. And another thing to note about the Hall Ranch area is that a lot of the fish that originate from Hall Ranch actually stay and rear in the Hall Ranch. And so that indicates an area that has a high degree of habitat complementation, meaning there's a mix of both spawning and rearing areas in the same place, which is different than having spawning in one place and then rearing in a completely different part of the basin. Um, now that you know how those graphs work, I'm gonna throw some more at you, but this is going through those different parentage assignment groups. So what you're looking at on the left panel are the natural origin, both male and female parentage, and on the right are the hatchery origin, male and female. And a couple of things to notice here are that all of the pure natural origin juveniles are basically dispersing down into the lower sections of the river. So there, you don't find any of those anywhere upstream, basically, of the restoration to Badger Flats area. And I think that's a really important result. When we start looking at the natural origin, the dam is always on the left and the sire is on the right. So the left panel are the natural origin females. What you would note here is that you are getting some of the natural origin females that are migrating up into higher parts of the basin and spawning with the hatchery origin males, who tend to be more exploratory from literature from other studies. Um, the dispersal process model, when we summarize all this information into that model that I described the methods for earlier, you basically get a graph that looks like this. So you've got dispersal probability on the y-axis and the distance from the red on the x-axis. And I think what I would want to point out here is that um, the this blue line are the natural, or sorry, this blue line is the restored sites, and the brown line are unrestored sites. And when we plot these two graphs side by side and scale the axes, they actually have an identical shape. So this brown line would look exactly like that line. And that just means that the probability of a fish moving further downstream decreases pretty rapidly as it starts to go downstream. And it's very improbable that a fish will migrate upstream. But when you actually scale these to a common y-axis, what you see is that restored sites retain those fish a lot better. So I think of them like being sticky. So restoration sites are sticky. They grab and attract fish that might be moving down through them. So, kind of getting into the discussion, I know that, for this meeting, we wanted a big part of it would be about adaptive management and about like what are the management implications. And I'm not going to stand up here and like give any management recommendations, but what I think I want to do is provide kind of a context or a framework for how you would use this kind of information to make conservation decisions depending on what your priorities are, because it's the basin, the collaborative effort out here that are actually coming up with what those priorities are. So first of all, we look at the dispersal patterns. This is the work that I just presented, determining where fish disperse to from their parent reds using this PBT approach. And then looking at the restoration and the supplementation. So understanding those strategies for the restoration, and then understanding the nuances of the dispersal patterns for the hatchery and the natural origin fish. Thinking about these ideas of riverscape complexity, so better understanding how the distribution of different types of habitat, whether it's good rearing habitat or spawning habitat, and how close or far those are, or whether they even overlap and serve the needs of multiple life stages. And then that leading to impactful management. So using all that information to make informed decisions. So that's kind of hand wavy and platitudes, but I want to go into some examples of how you would actually um, kind of think about that. So fish love restoration, but what are the priorities? So remember back to the natural origin crosses. So these are the juveniles that have two wild parents. If your management strategy is to prioritize juveniles from natural origin parents, you might just look at this area here and make some decisions about maybe putting more effort into restoration kind of efforts, or changing the riverscape to make it more conducive for connectivity between spawning and rearing habitats, or improving the rearing habitat down just in this area. But if your restoration priority is to benefit the highest number of juveniles overall, kind of ignoring whether it's a hatchery or a natural origin fish, then you would focus in on those areas that have a super high abundance. And remember, I said that these graphs don't do a good job of showing the magnitude of the fish, but there are a lot of fish. Most of the fish are spawning up in those Hall Ranch areas. Or do you have a smaller budget and you only have the ability to go in and like target individual restoration projects? And you might look at whether there were areas that were consistently lacking fish, and could you improve the habitat in those specific areas themselves? I wouldn't do this based just on a single year of study. I would want to make sure that these patterns kind of show up and repeat year after year. But that is kind of one approach that you could take if you were looking at, how to use a smaller budget to do restoration in smaller areas of the watershed. Um, so the summary of the management considerations is that Chinook salmon have a lot of overlap during their freshwater life stages. Supplementation and restoration need to consider both the adult and the juvenile habitat. And a lot of the restoration that we do is freshwater focused for Chinook, because we talk about things that are happening out in the ocean, but the management levers that we have are in the freshwater habitat. So we need to emphasize these concepts. Complementation and connectivity influence the spawning success, capacity, and survival of juveniles. Another thing that I didn't talk about is temperature. So this is the ISRP materials that were presented earlier, and I remember when I was working on the ISRP in my glory days at CritFic, working on the review for ISRP, and at that point, they have those confounding factors situation. And I think that we need to change the language a little bit because I don't think climate change is a confounding factor anymore. I think it's the major factor, the major driving force that we're seeing in these watersheds. So you look at the future projected summer stream temperatures for the western U.S. from the work that Isaac did with the Idaho Forest Service Lab. These streams are getting really, really hot, and like I mentioned before, the Catherine Creek doesn't have a lot of like outlet tributaries or cold water refuges that these fish can jump up into. Very different than the Middle Fork John Day, where fish could use those tributaries as kind of escape hatches. Um, there are limitations to our study. We had, there's some areas that we just could not access, so we couldn't access some private properties, or there's some gaps in that riverscape graph that I showed you where we don't actually know where fish or whether fish were distributing to those areas, limiting the juvenile capture opportunities. And the sample size for the natural origin fish was really low, and that's just a factor of the reality out there on the landscape. One thing that's not up here as a limitation that I mentioned earlier is that it was a single year of study, and so I think that it's in with this kind of a data when you only have 1 year, although it was a pretty representative flow year. Um, you have to, I think, kind of scale out and think about the implications that you would take away from a study like that at a broader scale and not focus in on like individual kind of micro reach habitat patterns that you see. So, kind of back to that picture of the theoretical watershed that I showed you, and just kind of thinking about what do we do differently now that we can figure out where fish are actually traveling through the basin? Like, how do we do restoration differently? And I think that's up for the partners to decide, but just kind of throwing out these ideas about restoring spawning habitat quality, looking at differences in hatchery and natural origin fish, and whether that's part of your program. Are you expecting introgression? Are you fine with that and happy with a high level abundance, or do you want to keep those separate and do restoration, that primarily benefits natural origin fish? And this is a really important tool for evaluating restoration projects that have been implemented. And what we showed in this analysis is that those projects are working if you consider the definition of working to be that they are sticky for those fish that are migrating through the basin. And then thinking about the bigger picture and conserving or enhancing the connectivity between different types of habitat rearing and spawning. And this should theoretically and hopefully practically help prioritize habitat complementation in the basin. And then Kayla always liked to talk about this at the very end of her presentation. One of the great things about being an advisor is that you get to work with students who have a lot of different perspectives. And Kayla had never worked on salmon before. Her background was in Colorado River desert fishes and in lamprey. And so when she came and started working on salmon, everything about salmon like blew her away. She was like really amazed, and that kind of freshness of looking at something that I look at every day from 20 years was really helpful to me. And she read Tom Quinn's book and came away with this idea that, wow, less than 1% of fish are anadromous. Even though salmon are part of our daily lives, they are a really rare and unique life history for fish out there in the world. So it's important to remember that. And those fresh eyes help me to keep track of that. Um, this, like I mentioned at the beginning, was very collaborative between all the organizations out here, probably others that are not on the slide, but we couldn't have done this without kind of coordinating the field work between all the partners, and a special shout out to the Bureau of Reclamation who funded the study and who kind of believed that we could do this kind of work. And Kayla is currently working on the um, uh, the version that we're going to submit to a journal. We have a draft. Her thesis is published, and that could be shared with anybody who is interested. And you can find that information at my lab at OSU. It's called the Fish Behaviorscapes Lab. The QR code points you to the web page. And with that, if there's any time, I think we're able to take questions. Yeah. Joseph.
Hey, Seth, great presentation. One of the other levers that you can pull is the hatchery supplementation program. The weir is very efficient, and so we have the ability to control what we put above the weir.
Yeah.
I'm actually curious in the male results, how many, so for if a male spawned with a female, did they spawn with one female or do they spawn with multiple females? And I'm curious to know, like, if there were any patterns between hatchery and natural origin males and the number of females they spawned with.
Really good question. And I'm going to do a dial of our end kind of a approach here. We haven't got that far into the Catherine Creek data yet, or maybe we haven't. I'm just not aware of it. But I know Joe Lemansky has looked at that, and Matt have looked at that in the Middle Fork John Day, and maybe they can speak to that for what we've seen in the Middle Fork and maybe early preliminary results for Catherine Creek.
Yeah, males, like, far and above tend to spawn with or have more mates than females. Females generally have 1 to 2 male mates. Males can range anywhere from 1 and, in the Middle Fork, I think the max we had was 7 females. In Catherine Creek, that was 9. So, and that's just a minimum. Like, there's also the potential that there could be other mate pairings that occurred that we didn't detect. Um, as far as the number of natural origin and hatchery origin females that a male might have mated with, I don't think we've pulled that information together yet. But um, in Catherine Creek in particular, I would probably hazard making any large inferences from that just because of the low number of natural origin fish that were actually sampled in Catherine Creek. Um, but that information should be forthcoming. So I guess to take away from that is that males can have a much broader impact on the population level and the combinations of, familial, anyway, so, yeah.
Yeah. One implication for that is, as you know, because you're connected to Emily's study, is like when you're thinking about what is the effective proportion of hatchery origin spawners in a river basin, there are these kind of new ideas about, well, if there's segregation among the hatchery and natural origin fishes, maybe effective PHOS is much smaller than like the PHOS that you would count going through a weir. But if males are traveling up and down that whole riverscape, then that kind of blows it out of the water. And you have to take the maximum range of male movement and consider that the spatial scale of a potential PHOS estimate. You know, Rich. Yeah. Or sorry, Winston has
I
the
got a quick question for actually you and Joe and others that participated in this. And first of all, thank you. And thank you for Kayla. Yeah, I know she's moved on, but I think this stuff is amazing and really brings a lot of light to fish in our basin. Um, how do we uproll what was done here? Very much like we've wired this basin for pit tags. Um, this data seems to be in this methodology seems to be very valuable. I mean, I sit there and I look at those graphs and it tells me what our projects are doing and or where we can improve. And as far as adaptive management, I will be talking about this tomorrow afternoon. But how do we uproll this? Because it obviously costs money. It was very labor intensive. Um, but it provides probably more information than I've seen a lot of studies in a very long time that are very applicable to what we do and the decisions we're making.
Yeah, thanks. I'll take a quick stab at that, and then maybe Joe has more ideas. But I I guess I'd answer that in two ways. One is um, connecting the finer scale labor intensive PBT analysis like we did here with what like Yuan is doing with NOAA, like just the overall PBT and different the relative reproductive success kind of stuff that you can get using PBT. So basically genetic samples of fish going up through a weir and then genetic samples of fish going down through a screw trap and looking at it that like really broad scale, and you're not learning anything about within basin dispersal, but you're still learning about differences in reproductive success between hatchery origin, natural origin, or different maybe different genetic compositions of the adults. and somehow, like, I can't picture in my mind exactly what it looks like, but making the linkages between the finer scale study and the broader scale study. And then I guess the second way I would answer that question is maybe rolling this kind of study out in different systems. Like we have the Middle Fork John Day, pure natural origin population, escape hatch tributaries. Compare that to Catherine Creek, that's warmer, doesn't have those escape hatches and has a supplemented populations. Maybe doing some like really cold water streams like the North Fork John Day or areas up in the Upper Columbia Basin, would be a good idea just to figure how general are these dispersal patterns that we're seeing because this is just the very beginning of what we know about it. You know. But Joe, thumbs up. Wow. All right.
Great.
Oh,
I'm gonna come to you, Rich, real quick, but we saw Kirsten.
I spend too much time with Winston. He mostly asked my question. But the I guess the other follow-up to that would just be kind of thinking in the same lines, like this is 1 year's worth of data, time intensive, all of that. But in terms of like using these data for a more specific like restoration planning process, like what is your thought on like how many years of data would be needed to have a good sense as to like wanting to use it for informing very specific, like where we do restoration activities, like what would that look like?
yeah. Um, Good question, and I'm kinda shooting from the hip here because I haven't thought out entirely what that process would look like, but I guess what I would do is go back to those slides that show, like, spatially where fish are originating from and then where they're going to, and then blur your eyes and look at where those general patterns are, even if you just have 1 year of data, because like, we have to make conservation and management decisions with the data that we have. We have to make some of them now. Adaptive management is all about like changing your perspective with new and incoming data. I I think that you could start right now with some of this data that we have and thinking about like because it's not a huge surprise to anybody that a lot of fish spawn in Hall Ranch. Now we know where a lot of them went in 1 year. Do a follow-up study, make sure that it's a consistent pattern. Um, but yeah, I'd love to chat with you more about that because I'm not going to be able to come up with a adaptive management plan on the fly, but it's a really good question. Yeah. Mm.
I think I have a mix of comments with the question. So my recollection is that there was actually in some years of high spawner abundance significant summer rearing below your lowest reach. Um, if that's the case, how might it affect your interpretation? And the other question is, I think when that happened, based on the summer pit tagging, that most of the fish that were in the lowest reach had no survival to smolt, to granite. And that leads me to another comment is I don't know that you can stop at the stage you're at and make totally sound recommendations because the fish have to survive at least to a smolt at granite. And you may have reaches that have high densities of fish, but the water temperatures are impairing their growth and making them not survive over winter or make it down to granite. And you have to factor that in your decision about what reaches you're going to focus your restoration on.
Yeah, really good comments. And the second question or the second comment slash question that you asked helps me articulate what I was trying to say earlier about connecting the bigger picture studies with the finer scale picture. So we'll eventually get relative reproductive success in terms of like fish going out through the screw traps, through all the catch points where fish go through. Um, we we didn't want to work that into Kayla's thesis because we thought it was a lot to ask a master student to take the data, but you could actually do a relative reproductive success study with the juveniles that were captured in the study. I think you'd have to like be okay with a slightly smaller sample size than you would want, but it's completely possible to do that. As far as the downstream, my um yeah, I think I remember that comment too, and there were some discussions about how far downstream we needed to sample for this study to make it viable. And I think there was some decisions that were made like, yes, we think that this is where we're going to capture the lion's share or like the 95% of the population. But can anybody else on the study comment on um, our decisions to not go further downstream, Polly?
Well, I mean, you sample the dried hashes, the lower half here, act that Grande at it is always limited, but I think it's gotten harder and harder over the years to get fish down there. Oh, sorry. Um, Presumably because temperatures are going up, or that's their hypothesis. So I think where we sampled fish for this project went as far down as we've caught fish in the last 10 years, but then again, we haven't looked farther down very often because we get so few down low. So, and based on that experience, I would guess that the study went as far down as they were going to find fish.
I'll just make a loud comment, but I'll repeat it. You might not have to, I mean, you might link up with the par pit tagging and survival rates as smolts to granite from that point, and not necessarily have to wait all the way for the adult to adult PBT results.
Yeah. Yeah, these fish were pit tagged, and so we would have the ability to do that.
Pull that survival.
Exactly. Yeah. For people online, Rich was suggesting that we could draw on the pit tag information, and we do have pit tags in these fish. Yeah.
Great. We might have time for one more question if there's one more.
Thank you. This is great. I have a question on Catherine Creek. If it's been supplemented for years, like it has, and you talk about natural origin and supplement fish, how natural origin are the fish that they're that you're designating as that?
Yeah. Um, Good question. You raise a really good point. This is a supplemented population. These fish are spawning with each other, and we even have results from that. I might do a dial a friend also and ask uh, Joe Feldhaus here if you can comment on how natural are the hatchery origin fish, or how hatchery are the natural origin fish.
Hatchery fractions in Catherine Creek are really large. I mean, we have a weir. We manage a number of hatchery natural fish are allowed to put up based on a sliding scale. Um, it's just a highly hatchery dominated stream. I I don't know. Rich could probably comment on this too.
So a natural origin fish is just naturally born. So they're, by definition, it's a natural origin fish. But obviously, if it comes from two healthy parents, it might have different survival characteristics, for example, than two natural origins there. But there's definitely been a lot of hatchery integration. But it's not an indication of major genetic change or anything like that. But as far as a natural fish, an active fish is an active fish just by definition. It's completely separate from a hatchery fish. There was a nagging gravel.
I think I made the mistake in my presentation of using the word wild, which is more like the fish that Emily Treadway is working with in the Wanahua River. Theoretically, those fish, have a genetically distinct genetic structure. And I don't think we're looking at the same type of thing. But you still see differences, even if a geneticist can't identify a hatchery versus a natural origin fish in a heavily supplemented population, you still see a reduction in the fitness of the hatchery origin fish that can last into the next generation. And newer studies are showing that that gets reversed or can get reversed in some populations, that fitness deficit actually decreases after fish has been spawning in the wild for a few generations. Yeah. Yeah. Yeah.
Great. Thank you very much, Seth.